Great. Well, it's really nice to meet you all. And yeah, thanks for taking the time to have a chat today. So, in terms of my background, I grew up about 50 miles south of London on the south coast in England near Brighton. And I went to school in a small village there. Um, my undergraduate degree was done at the University of Warwick. So I studied chemistry with medicinal chemistry at Warwick. And then I moved to the University of Bristol to do my PhD. And this was in a pharmacology department working on glutamate receptors. But I started to get interested in working on problems at the interface of biology and chemistry. Once I'd passed my PhD, I moved to Cambridge and worked with Andy Holmes's lab in the chemistry department, again, working on real chemical biology problems, working on phosphatidylinositol phosphate, so important intracellular signaling messengers. And then after 2.5 years in Andy's lab, I got my first independent position at the University of St Andrews, so right up in the north in Scotland. And I had 5 great years in St Andrews, and then we moved to Oxford in 2008, and we have been at Oxford ever since, having a good time doing research. We've got a lot of new results in the lab, so I'm struggling to work out exactly what to talk about. But what I'm planning to talk about will be some work that we've done on the TRIM proteins. So TRIM proteins are a really interesting class of proteins that contain bromodomains and PhDs. So as you know, we're interested in epigenetics, and we're interested in acetylation and methylation post-translational modifications. So the bromodomains read acetylation marks and the PhDs read methylation marks. So we've been looking, doing some fairly fundamental studies on the TRIM proteins to understand their preference for histone peptide binding. And then we've been trying to identify some small molecules that bind to the various domains. So that's, I think, one half of the talk will be about that. And then in the second half of the talk, we're going to talk about some bromodomains in the flatworm parasites, Schistosoma mansoni. So these cause schistosomiasis, which is a neglected tropical disease. And we have got the first X-ray crystal structures of some bromodomains in Schistosoma mansoni and got some high affinity ligands. And we're in the process of trying to correlate the effect of inhibiting these bromodomains or inhibiting their function on the parasite life cycle. So I'll tell you about some of the work that we've been doing in that. So we've worked for a long time on human bromodomains, but bromodomains in other animals are less studied, and it's really interesting to look at how they change the forms of their life cycle, throughout their lives, and it's very interesting to see how epigenetics affects this. Yeah, so we've got quite broad interests. So we do quite a bit of work on developing prodrugs that target compounds to hypoxia. So hypoxia is regions of low oxygen, which is found throughout biology, but we have a particular interest in the context of solid tumors. So as tumors grow, the vasculature struggles to keep up, and so you get regions of tumors that are hypoxic or have low oxygen, and that gives you a poor prog. It's bad for patient prognosis. But if you think about it in terms of the chemical environment of the tumor, it means the tumor's got a different chemical environment to the rest of the body, or much of the rest of the body, and therefore you can use this to target prodrugs there. So we've done some cool work recently where we have developed a hypoxia-activated prodrug of panobinostat, so an HDAC inhibitor. So again, epigenetics and acetylation. And we've shown in vivo that this compound is released selectively in the tumor. We think this could be an exciting technology going forward. And then related to that, we work on imaging hypoxia and actually imaging the redox environment as a whole. So again, we sort of think about the redox environment being the chemical environment of a cell. And therefore it's interesting to know what is going on in that environment. So we've got some, very nice work going on in trying to image different aspects of hypoxia and also reactive oxygen species. We've also got quite a lot of work going on now on bifunctional molecules, so a lot of people excited by this. And when I'm not going to talk about that, unfortunately, but again, that's something that we're very excited about at the moment. So we've got a very wide range of research interests. Yeah, so I think there's been a few things that we thought are cool. I mean, we're following with interest all of the PROTAC developments. Of course, the PROTACs in the clinic we're very excited by. We think this is really interesting. As a medicinal chemist, you sort of look at the properties of some of these molecules and think, wow, is that really going to be soluble, or is it really going to get into cells? And these things are working really well. So that's very exciting to see. We also like bifunctionals more generally. So I think there's been very cool papers looking at manipulation of other post-translational modifications using bifunctionals, so particularly phosphates. I think there's a very sort of big opportunity out there for people if you're thinking about bifunctionals in terms of modifying proteins going forward. So I think that's great. We also liked we have quite a wide range of papers presented at group meeting in our literature, and the Mark Levin skeletal editing by deleting nitrogen paper, the Nature paper, we thought this was quite cool. So this is kind of outside of our sphere of expertise, but it was great to see it presented and really, a neat new piece of chemistry, new disconnection that you could think about. And we thought that was very neat. So those are the things that have caught our attention recently. There's loads of other things as well, but those are those are the things that spring to mind as being really interesting. Definitely. I've got two bits of advice which I think are really important. So I think the first thing is always to work on things that you're really excited by and really interested in. And I think this is why I've ended up with such broad research group projects, that we're interested in a lot of things, and we work on things we're excited by. And then I think the other piece of advice is, when you're in the lab and when you're doing your experiments, try and think of everything as a hypothesis. And the success is testing the hypothesis. So if you do your experiment and your experiment works, then that's the success. Now maybe the result is what you want, and maybe it isn't what you want. But that's less relevant than testing it. Every time you do an experiment successfully, you should pat yourself on the back, because that's a real win, right? So I think that's an important piece of advice. It can get, it's difficult to see the big picture sometimes when you're doing research and you're very focused. So you just need to remind yourself every time you do an experiment, you've done a good job there, and that's important, I think. Chemists tend not to do hypothesis-driven research that much. We tend to think about, we've got to make this molecule. But if you can think about, my hypothesis is if I put this reaction on, I'll get this product out, then at least you've tested the hypothesis, right? So. Yeah, England played Germany last night. So we have a brilliant German postdoc in my group who's been wearing his Germany shirt. So we knew one of us was not going to be very happy this morning. So England have a terrible record of beating Germany. It's the first time we've beaten Germany in a competition in 55 years. So yes, I'm enjoying the Euros a lot at the moment. The next match is on Saturday against the Ukraine, so we will see how much I'm enjoying it after that. But yeah, it's been great, and it's been a tough year and a half, and so to see a little bit of sporting enjoyment and a bit of normal life returning, I think is a good thing. I've got two sons, so I like spending time with them and my family. Over the lockdown, we've done a lot of walking to try and keep them fit. But my enjoyment is distance running. So I like running half marathons and marathons. So Saturday morning for me is always a 13-mile run. So I like to go running with my friends. So go and do a half marathon in the morning, and then after that get on with the rest of the weekend. So and we're lucky in Oxford, you can get out into the countryside quite quickly and go to some nice places. So marathon and half marathon running is my fun. Yeah.